Mendel observed that traits usually assort independently during gamete formation. However, physical biology imposes a strict constraint on that rule. When two genes are located close together on the same chromosome, they do not sort independently. They move as one. This is genetic linkage. It is the physical tendency of specific genes to stay together during inheritance, passing through multiple generations as a single unit without separating. Scientists William Bateson and Reginald Punnett first identified this deviation in 1906 while crossing sweet peas. By 1910, Thomas Hunt Morgan established it as a core principle through his work with Drosophila. Because these genes are physically tethered, the number of linkage groups in an organism matches its haploid number of chromosomes. Drosophila has 4 pairs and thus 4 linkage groups. Humans have 23. This arrangement directly restricts the genetic variability available to the offspring. Linked alleles organize in 2 ways. In the cis phase, dominant alleles share one chromosome, while recessive alleles share the other. In the trans phase, the arrangement is mixed. Each chromosome carries one dominant and one recessive allele. In either case, linkage prevents the traits from mixing. To restore variation, nature uses a physical mechanism to break these tethers and shuffle the genetic deck. That mechanism is crossing over. It occurs during the pachytene stage of meiotic prophase 1. As homologous chromosomes align, their non-sister chromatids physically cross over at a point called a chiasma. The strands break at precise locations and then reunite with opposite partners. Crossing over serves as the biological counterweight to linkage. It generates the recombinant gametes that provide the variation required for evolutionary survival. Crossovers are categorized by their source. Germinal crossing over happens during meiosis and is universal across sexually reproducing species. Somatic crossing over occurs during mitosis, as noted in the fungus Aspergillus, but it is extremely rare and lacks evolutionary significance. We classify crossovers by the number of chiasmata. A single crossover yields a 50-50 split of parental and recombinant gametes. But in a double crossover, a second swap undoes the first, producing only parental gametes and hiding that a crossover occurred. The further apart genes are, the higher their crossover frequency. We calculate this recombination frequency by dividing recombinants by the total. 1% equals 1 centimorgan. Because map units are additive, if A to B is 5 and B to C is 10, A to C must be 15 centimorgans. By combining the results of multiple 3-point test crosses, scientists can determine the exact linear order of every gene on a chromosome. Pinpointing gene locations provides the blueprint for modern agricultural breeding. It allowed researchers to isolate the high-yield traits that defined India's Green Revolution. Linkage keeps genes together, reducing variation, while crossing over breaks them apart, increasing it. Linkage strength is inversely proportional to distance. Crossover frequency is directly proportional. Understanding this push and pull provides the logic to solve chromosomal mapping.